I'm Elisa, and we've been talking about the circulatory system, in particular, the blood. And this time we're going to talk about the hematocrit and some basics about plasma in the blood. Okay, let me get a green pen. Okay, so general components and properties of blood. So the idea is we need to look at well, essentially this. Let me show you on this one. This is a little bit better. So we've been talking about how blood is a connective tissue and that it's a connective tissue that is it's liquid primarily. And that's the way that it is a connective tissue is that it flows through the body and it brings nutrients, it brings oxygen, and it takes away things like CO2 and it takes away things like waste. And in order to do that, it has to have a certain composition. We talked about the idea that the extracellular matrix is really primarily this blood plasma. And so if this is like 100%, right, like this is the 100% of like just a little vacutainer that you would have taken of blood, you really don't need that much. In somebody who's well hydrated and healthy, we're expecting it to be around a little bit more than half is like the liquid portion of blood or the plasma. Um, and then about 45% is usually the red blood cells, right? We started looking at those. We'll get more into those later on. And then we have this other area right here, which is called the buffy puffy coat. And it's like this white stripy area. And what those are the white blood cells, leukocytes, right? So lucid cells, remember? And they shouldn't be too much of a percentage of the whole blood if someone is not struggling with something like, possibly like an immune response, right? You need some more white blood cells, and so that would be higher. Or in some certain cancers that, like leukemia, for example, would have a higher percentage, and that's, a diagnostic tool. Other things that I want to show you that is kind of not useful in this one is the blood plasma here, right? Normally, it would be like in a healthy individual, like a really light yellow, actually. Like you want it to, it's kind of like, remember, this is, well, remember, this probably before your kidneys chapter anyway, your urinary chapter. Um, the blood plasma that's filtered out is typically going to be what primarily composes one's urine, for example. And so when we look at it, we want to see it kind of like a really light, faint yellow, right? Not a really dark, heavy yellow, um, for example. And so that's what we'd be looking for the hematocrit. And so now that you know what we do, we take a little sample, and then what we do is we use G-forces. So we centrifuge it down. And this little machine, if you've never used one, it's kind of fun. You pop it into this, see where that little blue dot is? You pop it into the container, and they kind of lay and they stick out the sides, right? And then this thing circles around really fast, like a washing machine on the spin cycle. And when it does that, it pulls the heavier part of blood down low, and then the lighter part, right, the more just watery plasma up high. And so the heavy blood cells that are saturated with iron, theoretically, would be going lower than, say, the plasma, and then the middle weights or the leukocytes would get stuck in the middle. And that allows us to determine the percentages, relative percentages of blood plasma to red blood cells to white blood cells. Okay, let me kind of pop back. So we know now about centrifugation. So that's that spinning motion, right? That just like a spin cycle on your washing machine for your clothes at home, right? So it's all the heavier components go out to the bottom. So your erythrocytes, the red blood cells, as we saw, are the ones that are heaviest and they settle first, right? There they are. They're at the bottom. Okay. And so they want to make up around between 50 and 55% of the total cell volume. The lower it is, that might mean a few things. I mean, it could mean that someone is like overhydrated for one reason, and for one example, but then another reason, it might be a sign of anemia, right? Or the inability to produce enough red blood cells, or possibly lysis of red blood cells. So you don't really want to get super low. I would think that's actually on the low side. So when we look at a hematocrit, we're actually talking about this thing called the packed cell value. So you might see PCV, right? So that would be the same terminology. White blood cells and platelets, as we saw, they kind of end up in the middle, right? So they are right here in that puffy buffy coat. Okay, so they should be about a little bit less than around 1% of the total volume of the container. And, like we said, it's with a puffy coat. And it's, the hematocrit, just so when we're measuring this, there's not some kind of crazy, super brilliant science to it. Let me just show you. Like what we would do is we get a pencil working. If you took a ruler here, right, and you said this was like, however many, like 5 centimeters, right? Like this is 5 centimeters. There's 1 centimeter, 2, 3, 4, 5. That should be more of a 4. That looks like a 4. Okay, we're gonna look at it like right at around here, right? So you literally just measure it, okay? And some of you, that's what we do in the labs. So if you're in our A&P labs, we will be doing that, okay? And then the last value is the plasma. So you want it to be relatively high, right? So you want you want folks to be hydrated. People that are coming in with lower volumes of plasma. So if they have a hematocrit, right, and you pull it and you spin it, and like you're seeing like a like a really high ratio of red cells, for example, to plasma, that could mean a few things. And one of those things is dehydration, actually. And some of those, here's this puffy, buffy coat right here, right? This could be dehydration. This could be, overproduction of red blood cells, which we'll look at a little bit later. So they tell you some important things about the status of your patient right away. Okay, the thing about plasma is it's not like pure water or anything. It's actually a bunch of stuff. So it's a mixture, right, of obviously water, different types of proteins, which we'll look at, functional proteins, especially for clotting and for communication. They have hormones, speaking of communication, some of these dissolved gases, right? Um, like CO2 definitely does travel via plasma. Some nutrients, electrolytes, which are, anything from just small ions to larger ones, right? To larger molecules, especially ionized ones. And then nitrogenous waste, the ammonia that ends up eventually in the like urine and other forms also. So here we go, just to get on that process. So we take whole blood, right? Regular old blood draw. We centrifuge it, right? Put it through the spin cycle. It's going to split into two parts, the formed elements. So those are our RBCs, red blood cells, erythrocytes, that's, red blood cells. And then the plasma, and then also, like buffy coat, buffet, right? Coat. And so the plasma is going to tell us a little bit more also about um, coagulation. So oftentimes you get these and there'll be like a little bit of something down here in the bottom in the tubes before you do your blood draw. And oftentimes that'll help prevent immediate coagulation of the blood because there are a lot of different proteins that are going to stimulate that response, or they're going to start to settle, or they're going to start to become active. Okay. And so that can be pulled into two different categories. Number one are going to be removal of the clotting proteins or isolation of them, and then the serum proper, right, which would be outside of those proteins, all the more liquidy parts. Okay, blood plasma. We talked about it's the liquid portion of blood. It's got some dissolved elements in it as well. The serum and the plasma proteins, oops, sorry, plasma proteins. I didn't mean to get you crossed out there. We'll go. A little better. Bloop. Okay, it's the liquid portion of blood, and it'll be divided into serum or plasma proteins. So the plasma proteins are all of these globulins, fibrinogens, albumins. Many of them are produced by the liver. The liver makes so much stuff, you guys. And when we get into digestive, you're going to be like, wow, here is the, I don't know, biological medal of honor. You've done so much work for us, liver, and you're so diverse in the things that you can make. For the most part, so the globulins, those are plasma cell based. So serum itself is going to be everything that the blood clots and solids are removed. And it's identical to plasma except for primarily this fibrinogen. And so fibrin, think of it makes fibers, right? And so we use these a lot in clot formation and scars and all kinds of stuff throughout the body. And then gen is derived from the term genesis, right? No, yeah, I should know better. Sorry, I don't have a degree in spelling. But anyway, speaking of which, so like it's where the word gene actually derived from because it means the beginning, right? The start of something. So it's like the start of these like fibers. So it's kind of like just the molecule that's like maybe holstering and wrapped them up, right? Tight would be fibrinogen. And then when they are loosed out, they become like fibrin and all that. And that's one of the important components of like clot formation and healing. Okay, so the three major categories of plasma proteins, albumins, a lot of these, so they're tinier, they are the most abundant, and what they do is they help maintain blood viscosity and kind of like a balance. They're like little buoys in the pool. We use them a lot in molecular biology just to kind of helps separate and evenly distribute ions throughout a solution so we get a more like dependable reactivity. And they also contribute to osmolarity in that way because right we're dividing and we're kind of separating out all of our ions so we're not having clusters. um, they obviously are going to influence blood pressure in that way and balance as well. So globulins, think of like they glob onto stuff and sometimes it's also each other, but what they do is they provide immune system functions, right? So immune in itself is usually talking about proteins that are going to like stick to something or recognize an antigen. And so there's alpha, beta, and gamma globulins, and we'll get into that much later. We talked also already about fibrinogen, the precursor of fibrin threads, remember from scarring or from clot formation, right? They're like the duct tape that floats around, and they help form, in this case, blood clots. And we see it a lot of other places in the body. So just heads up on that. Okay. Well, that is a few minutes on the hematocrit and some information on the plasma of the blood. And we'll definitely be continuing, but I like to keep these videos super short and sweet. So I hope you're having a lovely evening, and I will see you all in part 4. Bye.